So you see the citric acid cycle here. We had just finished talking about that. Again, think of that as one of the most important mining steps of cell respiration because it liberates or frees up a large number of electrons. You see that as NADH here, FADH2, and more NADH. And all of those electrons go over to our transport chains, which is in the power plant step, oxidative phosphorylation, which we're going to talk about next. So, again, broad overview. We've talked about glycolysis first. We talked about the prep reaction. We talked about citric acid cycle. Now, we're going to spend the rest of the chapter talking about oxidative phosphorylation, which produces a large amount of ATP, the most ATP of any of these steps. All right, so following glycolysis in the citric acid cycle, the potential energy from our food molecule that we can now use to build a lot of ATP is bound up as two electron carriers, NADH and FADH2. And those carrier molecules will now come into the transport chain, which you see here, and they're going to dump those electrons off at the top of that transport chain. NADH at the very beginning of it, FADH2 slightly further downstream. The difference doesn't really matter. The point is, those electrons will move down the chain from protein to protein. As they do that, they lose energy. They're going from high energy when they come in to low energy when they come off the chain. And so, because they're losing energy, that tells you that there must be a way to take that energy and store it up because the cell is going to use that energy to build electrons, right? So why is it that these electrons are moving down that chain in this direction? It's because oxygen is sitting at the bottom, and oxygen, again, being electronegative, pulls those electrons down the chain, forcing them to give off their energy. This is why you have to breathe oxygen. If you don't have oxygen in your cells, your electron transport chains don't run, and you can't make enough ATP for cells to survive, and so cells die. That's what asphyxiation is. All right, so what is this? How is this energy that these electrons are giving off going to be used? Well, notice that we have these active transport pumps, number 1, number 2, 3, and number 4 here. As I said, these are active transport pumps. Because they're active transport, that tells you that they're using energy to pump something. What are they pumping? Protons, H+. They're going to pump it across the inner mitochondrial membrane. Those protons are going to build up in high concentration on one side of the membrane, just like water behind the dam. We've talked about the hydroelectric power dam analogy. Water built up behind the dam represents stored potential energy. It flows through the dam. Potential is converted to kinetic motion. That is then converted by the machinery in the dam to electricity, as we talked about. The same basic thing is going to happen here. Okay. All right. So the transport chain that you see here then is located in the inner mitochondrial membrane. Remember, mitochondrion is shaped sort of like a kidney bean. Outer membrane is smooth. You have an inner membrane that is looped and folded like such. And the transport chains are in that looped and folded inner membrane. So the more that membrane you have, the more transport chains you have or copies of it, and the more ATP can be produced ultimately. All right. So these proteins that are embedded in the membrane that act like steps in the staircase that the electrons fall down, many of them belong to a class of proteins called the cytochromes. You can see that abbreviation right there, CYT. It's not really important, but it's just a class of proteins that you find that are all sort of working together to do this one thing, which is force these electrons to give off their energy. And again, we have oxygen at the bottom, pulling those electrons down that chain. Okay, so the electron transfer in the transport chain causes the proteins, the active transport pumps, to pump protons from the mitochondrial matrix, so this is the matrix, to the intermembrane space, which is here. Sorry about my writing. That's kind of weird with the Apple Pencil compared to a chalkboard. Okay, So protons get pumped to the intermembrane space, and again, that is where they're going to build up in high concentration. This is like the water behind the dam. Okay? All right, so if we have a high concentration of protons on this side of the membrane, what does nature like to do to gradients like that? Eliminate them. Those protons are going to want to flow back across the membrane to the matrix. But they're ions. They can't get it across the membrane directly, so they need to be able to move across some sort of protein acting as a channel. And that's what this protein is. This protein is called ATP synthase. So what does it do? It synthesizes or makes ATP. All right, let me change colors here. So here we have the mitochondrial intramembrane space. That's where we have high proton concentration. Here we have the matrix. This is where we have low proton concentration. So the protons are going to want to flow in this direction down their concentration gradient, just like we saw in the diffusion chapter. Okay. All right. Well, they can't get through that membrane directly, so what they do is they drop into the ATP synthase enzyme. So, excuse me, as the potential energy of protons and built up behind a dam is converted to kinetic as they flow in, that kinetic energy actually causes part of the ATP synthase enzyme to rotate. The rotation of that top part, called the stator, basically, which you see right there, then causes the catalytic knob, let me change colors again, catalytic knob here to rotate. When that rotates, it exposes active sites that accommodate ADP plus phosphate, and those then get converted by the enzyme to ATP. So if you're adding phosphates to ADP, you're building something, correct? That means you have to have a source of energy. Well, what is the source of energy? It is the protons flowing from high concentration to low. Just like water through the dam, same thing, it gives off energy. That flow of protons from high concentration to low is exergonic. As we've talked about, that means it releases energy. Well, the enzyme captures some of that, exposes that energy, causes the enzyme's active sites to get exposed, ADP and phosphate fit in, and they're converted to ATP. All right, so this is the basic mechanism of oxidative phosphorylation, at least in terms of how the energy of the electrons is converted to a proton gradient, which you see here, and that is then used to build ATP, just like water flowing through the dam, but it's protons flowing down their gradient. All right, so this is the whole thing then put together, oxidative phosphorylation. So the whole thing here is oxidative phosphorylation, made up of two parts, electron transport chain and chemiosmosis. All right, let me change colors again. Okay, so here we have the electron transport chain. You have NADH, FADH2 coming in at the very beginning, dumping off their electrons. The electrons move down the chain, as you see here. So they're moving this way down the transport chain, all right, giving off their energy, which is then used to pump protons across the membrane, as we said. So we have a H plus gradient here in the intramembrane space. Protons flow back through ATP synthase, give off their energy, and ATP synthase produces a large amount of ATP in the way I talked about. So the transport chain liberates the energy from the electrons. That potential energy is converted to the energy of a proton gradient. ATP synthase allows those protons to flow back through the membrane down their gradient. Their energy is given off, and that's used to make ATP. That specifically is chemiosmosis. So oxidative phosphorylation is made up of the electron transport chain and then chemiosmosis. All right, so this energy that we then have stored up as a proton gradient is referred to as a proton motive force. The word force implies that energy is stored in some way and can be used to do work. Here it's stored as our proton gradient, the water behind the dam, so to speak. Okay? All right. So, overall then, during the whole process of aerobic cellular respiration, energy has flowed in the following sequence. It's gone from glucose to then NADH and FADH2 to the electron transport chain, stored temporarily as a proton motive force, and then finally to ATP. So after all that is said and done, about 34 to 36% on average of the energy in the original glucose molecule has been transferred to ATP during cell respiration, making about 32 to 34 ATPs, something like that. Don't forget, we had 2 ATPs made by substrate level phosphorylation in glycolysis. We had 2 more made by the same process, substrate level phosphorylation in citric acid cycle, and then we get about 28, 30, something like that made by chemiosmosis right here in oxidative phosphorylation. So in total then, we get about 32, 34 ATPs. The number varies, but that's usually what you see in the most recent editions of the textbooks. All right, so here it is in summary then. Glycolysis produces 2 ATPs through substrate level phosphorylation. You get 2 more in the same way through citric acid cycle. What's more important though are the electrons coming out as NADH, okay? And those electrons, as you see, along with the electrons from the other parts of the process, end up over in oxidative phosphorylation. They move down the transport chain. Chemiosmosis uses that energy to make ATP. At the end of glycolysis, we end up with 2 pyruvates that you see here. They enter the mitochondria. Remember, the PrEP reaction converts the pyruvate to acetyl-CoA, which is then compatible with citric acid cycle. That process frees up more electrons, which go over to our transport chain. All right, citric acid cycle breaks down the acetyl-CoA now down completely into CO2, so it's converting organic carbon to inorganic. At the end of the citric acid cycle, all the original 6 carbons, organic carbons and glucose, had been converted to CO2. But you see, citric acid is really important because of all the electrons it pulls out. That's a large amount of potential energy right there that then gets funneled over to the oxidative phosphorylation or power plant step. So because of all these electrons moving over this way, we get a large amount of ATP, okay? All right, so final thing here is to talk about the versatility of catabolism. So catabolism is what we just talked about. It's any process that breaks down food molecules and extracts energy. And for the most part, we focused on aerobic cellular respiration, okay? It's a very versatile process. We've talked about it in terms of, running basically a sugar molecule through. So we've talked about carbohydrates going through and glycolysis, a PrEP reaction, citric acid cycle, oxidative phosphorylation, all that. But we could just as easily have talked about a protein molecule moving through. Proteins are first broken down to their component amino acids, and the amino acids can be injected at various different points along the way, depending on which amino acid we're talking about. Some of them are converted directly to pyruvate, others directly to acetyl-CoA. Others can drop directly into the citric acid cycle, but either way, the energy of the protein ultimately is funneled into oxidative phosphorylation, where a lot of ATP is produced. One thing about protein metabolism is that it produces ammonia because amino acids have amino groups. And so ammonia is very toxic. That's one side effect, or downside rather, of protein metabolism, is that it produces a lot of ammonia, which we have to either get rid of quickly or store up in a less toxic form. And mammals primarily convert ammonia to urea. That's what one of the primary ammonia-containing components of mammal urine is. It's a safer way to store it up. All right, fat molecules over here on the right. Fats, very energy rich. On average, a gram of fat contains twice as much potential energy as a gram of protein or sugar. Fats are first broken down into glycerol and their component fatty acid chains. The glycerol can go directly in as G3P. So the energy of that glycerol funnels directly in as G3P, which, if you recall, is the end product of the investment phase of glycolysis, and it gets funneled into the process that way. Fatty acids are used to converted directly to acetyl-CoA, so their energy is funneled in that way, and again, finally, the electrons go to oxidative phosphorylation. So the point here is that aerobic respiration is very versatile. It doesn't just rely on sugars. We can eat a piece of pepperoni pizza, for example, and you get obviously carbohydrates from the dough or the crust, but you also get fats and proteins from the pepperoni, from the cheese. We can take advantage of all that. You can even funnel nucleic acids, DNA and RNA, through this, although it's not shown because that's in our food as well. So it's an extremely versatile process, which means we can get a lot of energy out of our food. Okay, very last thing to talk about in this process is how aerobic respiration is regulated. What are the feedback mechanisms that control this? Remember, we talked about feedback inhibition at the end of chapter 8. Basically, it's a process whereby the cell sort of monitors the internal environment or some aspect of it and uses that information to either speed up or slow down a metabolic pathway. Well, this is a pathway, and if you're sitting around not doing much, your rate of cell respiration is relatively low. If you're up being very active, it's much higher because you need more ATP. So our cells have to be able to sort of switch this process on, speed it up, slow it down throughout our day as our energy demands change. All right, so primary compound that's concentration is measured in this in cell respiration to regulate the rate of it is ATP. If ATP concentration begins to drop, respiration speeds up. When there's plenty of ATP around, respiration slows down. So the ATP concentration itself is something that's measured by the cell as an indication of when to speed up and slow down respiration. And control of catabolism in this way is based mainly on regulating the activity of enzymes at strategic points in the pathway. Typically, enzymes more toward the beginning of pathways are the ones that are kind of acting like these control switches, right? So let's take a look. So this figure is showing you basically the way in which cell respiration is regulated. So again, we start off with the glucose molecule here. It's being broken down through glycolysis, which you see here. Again, we have prep reaction. Oops, sorry about that. Prep reaction right there, citric acid cycle, and then oxidative phosphorylation like we've talked about. Okay, so what happens if your ATP concentration is high? Let's say you've been very active, your rate of respiration is very high, then you become inactive, you sit down after exercising, let's say. Well, there's a sort of a time lag there where your ATP concentrations begin to build up because now all of a sudden you're not using it very quickly, but you're still producing it quickly as if you're exercising. So your ATP concentration begins to build up, and ATP binds to that enzyme, phosphofructokinase. You should know that one. It's one of the most important regulatory switches in aerobic respiration, cell respiration. It's toward the beginning of glycolysis. When ATP binds to phosphofructokinase, it shuts the enzyme down, which means everything downstream slows down. So your ATP production goes down. You're still using your ATP, although at lower rates, so eventually your excess ATP supply diminishes, and the cell's only option then is to pull the ATP off phosphofructokinase to use it right here, and then that reactivates the enzyme, so everything downstream speeds up again. All right. Now, what if your ATP concentrations are low? All right. If they're low, then that means you don't have enough. That means that the cells need to increase their rate of cell respiration. So first thing that happens is that, well, the cell pulls a phosphate off of ATP. That then makes ADP. Well, if you convert all your ATP to ADP because you're not making ATP fast enough, the cell's only possible action then is to then pluck another phosphate off of ADP, converting it to AMP. Well, you see the AMP right there then stimulates that enzyme, phosphofructokinase. So again, we see this enzyme acting as a switch, right? In this case, AMP binds to it, stimulates it, and speeds up everything downstream, thereby producing more ATP. All right. One other thing I forgot to mention, citrate. Citrate, which if you recall is the first organic intermediate citric acid cycle, why it's called citric acid cycle, if that begins to build up in concentration, it can inhibit the same enzyme, phosphofructokinase. So why would citrate begin to build up? Citrate builds up if your ATP concentrations build up. Think of this as sort of backflowing into the system. Citrate is still being produced because acetyl-CoA is coming in, but it's not being processed by the citric acid cycle because the cycle is slowing down, and that causes a buildup of citrate, which is sort of a secondary way of shutting down phosphofructokinase. It's sort of a two-pronged attack basically on the enzyme when you have too much ATP around. There are two distinct ways to shut it down to make sure it's shut down quickly so the cell isn't wasting resources and building ATP when it doesn't necessarily need to. All right, so last thing I have here, assuming this video works, is an animation of, a Bioflix animation of cell respiration. Hopefully this will work. If this does work, you're going to see it kind of glosses over glycolysis and citric acid cycle, but it does a really good job of showing you the electron transport chain. So let's see if we can get this to work.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy in fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.